Hello and how are you again? My name is Winnie Barawa. Welcome to our session on mitosis. So mitosis is one of the types of cell division and the main types are mitosis and meiosis. However, in this video, we are going to focus on mitosis. If you're new, this is a channel that focuses on content relating to healthcare ranging from lessons, lectures, and expert interviews. Keep with me as we explore more on this particular topic. So mitosis is a type of cell division and it is described in terms of four phases. We have phase 1 that we call prophase, followed by metaphase, anaphase, and telophase. And these are highlighted description or illustrations of how these stages change. So we have prophase, the first one, followed by metaphase, anaphase, all the way to telophase. So exploring prophase, because this is the first stage of mitosis. In the prophase stage, being the first stage, we are told this is the stage where the chromosomes begin to condense and they become visible. The spindle fibers emerge from centrosomes or the centriole areas here, and we form stars or also what we call asters. So asters extend from the centrosome matrix around the centromeres. The nuclear envelopes breaks down and the centrosome starts to move towards the opposite poles. So what is happening here? The chromosomes are condensing and exposing the sister chromatids in the nucleus. We are forming asters or stars, and this is activation of the centrioles, and these centrioles will be moving in the opposite direction, that is in the north pole. One will be in the north pole and another will be in the south pole. And from these asters, you will now produce the spindle fibers. And these spindle fibers are very, very important because they will be existing in form of microtubules and microfilaments that are very important in the cell division process. So by the end of prophase, we expect that the nuclear envelope has disintegrated or it has broken down. The centrioles or the centrosomes have moved in the opposite direction in the poles. We have one in the north pole here and another one on the south pole. And from them will originate the microtubules. And these microtubules are very key because they will be attaching to the kinetochore. Kinetochore is the joining area for the chromatids of the chromosomes. So this will be marking the end of the prophase stage. Then in the metaphase stage, we are being told that in the metaphase stage of our mitosis, the chromosomes continue to condense. However, the kinetochore are appearing on the centromere and mitotic spindle tubules are attaching to these kinetochores. So the chromosomes end up lining up at the metaphase plate and each chromatid is attached to the spindle fiber that has originated from the opposite pole. So as you can see at this particular time, our cell now has what we call longitudes, which are formed by the spindle fibers, and then we have the equator, which will help us in establishing the metaphase place. So the metaphase plate is the structure that forms here after we have aligned our chromosomes or our chromatids along the equator. So once we have aligned these chromosomes along the equator, we form what we call the metaphase plate. So the metaphase plate just confirms the proper arrangement of the chromosomes along the equator in the metaphase. Basically, that's the summary of what happens in metaphase. There's condensing of the chromosomes, the appearance of the kinetochores at the centromeres, the spindle fibers attaching to the different sides of the kinetomeres, and the chromosomes lining up at the metaphase plate. And the other stage is the anaphase. And in the anaphase, we are talking about splitting of the sister chromatids into two daughter chromosomes, and the centromeres will split into two. So the sister chromatids, now we shall call them the chromosomes, will be pulled in the opposite poles or into the opposite ends of the cell. So the certain spindle fibers will begin now to elongate the cell, because as you're pulling the chromosomes into the opposite side of the poles, we shall be elongating these cells and making it a bit longer in terms of length than the original mother cells. So the anaphase stage is the splitting stage of our mitotic stage. And once the anaphase stage has successfully taken place, we transit to the telophase stage. And in the telophase stage, we are being explained that the chromosomes have arrived at the opposite poles and they're beginning to decondense. Because this process of decondensation happening, the nuclear envelope, the materials will start surrounding the materials around the chromosomes, and our nucleolus will reappear. So as the nucleoli reappears, the mitotic spindle will break down, and this will indicate the starting point for the mitosis that is ending. So this is how the telophase is happening. So the chromosomes have arrived in the opposite poles and they are beginning to decondense. So the nuclear envelope materials will start now surrounding again, and the nucleoli will reappear here, as you can see. So as the decondensing is happening, this is simply telling us that the DNA material is starting to twine itself around the histones again and going into the storage stage until needed for further cell division. But I want us to note that there's a ridge here that is coming up. In this ridge, we are calling it the cleavage furrow. As the telophase stage is moving to the last part, we normally initiate the cytokinesis, and that is division of the cytoplasm. So this is cytokinetic stage. So as the mitosis stage draws into close, this is now the telophase stage. Cytokinesis will start to help us complete the division of cell into two daughter cells, and that is the diploid cells, which have similar chromosomal number just like the mother cells. So a contractile ring of peripheral microfilaments will form at the cleavage furrow, and then it will squeeze the cell apart. So each is smaller and has less cytoplasm than the mother cell, but they are genetically identical to one another. So here is our cytokinesis happening. A contractile ring has happened at the cleavage furrow, and our two daughter cells will be produced. They will look smaller than the parent cell, but they'll have the same genetic characteristics as the parent cell. So here is a summary. I wish us to remember that mitosis is part of the stages of the cell life cycle. So because it's the part of the stage of the cell life cycle, what happens is when the mother cell undergoes DNA replication, it will double its DNA chromosomes or the chromosomal numbers. So it will end up with sister chromatids with similar characteristics here. However, during the cell division of mitosis, we shall separate these sister chromatids, whereby we will now have chromosomes into the daughter cells. So the daughter cell will end up with similar chromosomal number, just like the mother cell. So if you look here, this is the mother cell during the cell life cycle. It had this particular number of chromosomes, just using it as an example. And then during DNA replication, the chromosomes double because it created copies of itself. Because it created copies of itself, these copies go through the mitotic cell division and they will pull apart those chromatids into creating chromosomes that are equal number onto the two daughter cells. However, if you look at these two daughter cells, they have the same chromosomal number just like the mother cell. And so this is a summary. After the interphase stage has completely taken place, we will transit it to the mitotic phase. And in the mitotic phase, we have prophase, metaphase, anaphase, and the telophase stage. So once the telophase stage is successfully completed, we go through cytokinesis and we have two daughter cells which can either wait until the cell is ready to go through the cell cycle again till reproduction or division, or the cell can go through the G0 stage where there is no cell division and it will just do its normal activities without potential to divide. So thank you for being with me all the way to the end. Remember to share these videos, click on the subscribe button and comment, and remember also to like. Thank you so much and see you in our next lesson.